Beneath the threshold of human sight, an invisible billion-year war rages in every drop of ocean water, every handful of soil, and every biological ecosystem on Earth. It is a relentless conflict of total annihilation between bacteria and the parasitic viruses that hunt them, known as bacteriophages. The stakes are absolute, dictating the survival or extinction of microscopic lineages on a moment-to-moment basis. For decades, human science viewed bacteria as relatively simple organisms, lacking the sophisticated adaptive immune systems found in higher animals. We assumed they survived viral onslaughts purely through rapid reproduction and random mutation. But nature harbors profound secrets in its most ancient architectures. The ultimate weapon of this microscopic war, forged in the crucible of evolutionary pressure, would eventually be unearthed by humanity, entirely by accident, profoundly altering the trajectory of our own species. The genesis of this biological revolution did not begin in a gleaming, state-of-the-art genetics laboratory, but in the blinding, sun-scorched salt marshes of Santa Pola, Spain. In the late 1980s, a young microbiologist named Francisco Mojica was studying microbes that thrived in these extreme, hypersaline environments. He was specifically focused on an organism called Halophorax mediterraneae, trying to understand how its genetic code allowed it to survive in conditions that would instantly dehydrate and kill most other forms of life. At the time, sequencing DNA was an agonizingly slow, manual process. Researchers had to read the genetic letters, adenine, cytosine, guanine, and thymine, one by one, deciphering the fundamental code of life through sheer, tedious repetition. During this painstaking work, Mojica noticed an anomaly that defied biological logic. Deep within the microbe's genome, he found a bizarre, stuttering pattern. There were short, identical sequences of DNA that read the same forwards and backwards, known as palindromes. But strangely, these identical sequences were separated by equally short, entirely unique stretches of genetic code. It was a pattern of repeat, spacer, stretching across the genome. In the hyper-efficient world of microbiology, where organisms routinely jettison unnecessary DNA to conserve energy, this repeating genetic gibberish made absolutely no sense. Why would a microbe dedicate precious resources to maintaining such a strange, repetitive structure? The mystery deepened as the scientific community largely dismissed these sequences as mere junk DNA, a meaningless artifact of evolutionary history. But Mojica was captivated by the sheer improbability of the structure. His conviction grew when he began scanning the emerging, primitive genetic databases of the 1990s and found these exact same repeating architectures in entirely unrelated species of bacteria and archaea separated by billions of years of evolution. Nature is ruthlessly pragmatic. If a genetic structure is conserved across vastly different branches of the tree of life, it is not junk. It must serve a fundamental, indispensable purpose. For nearly a decade, the true function of these arrays remained locked away, constrained by the limited genomic data of the era. Mojica became a genetic detective, spending years cross-referencing those unique spacer sequences against every newly published fragment of viral and bacterial DNA. The work was isolating, often unsupported by major funding grants, driven solely by the pure, obsessive curiosity to understand a biological anomaly. He was tracing the faint outlines of a microscopic mechanism that no one else believed existed. The monumental breakthrough arrived in August 2003, hidden within a massive spreadsheet of genetic alignments. Mojica realized that those unique spacer sequences, the random bits of DNA trapped between the repeating palindromes, were not random at all. They were exact genetic matches to the DNA of known bacteriophage viruses. The implication was staggering. The bacteria were not just harboring junk. They were keeping a highly organized, chronological archive of the viruses that had previously attacked them. They were storing genetic mugshots of their enemies. Mojica and his colleagues eventually named this system CRISPR, standing for Clustered Regularly Interspaced Short Palindromic Repeats. This revelation fundamentally shattered the existing biological paradigm. It proved that bacteria possessed a sophisticated, adaptive immune system. When a virus injected its deadly genetic material into a bacterial cell, if the bacterium survived, it would somehow snip a tiny fragment of the invader's DNA and integrate it into its own CRISPR array. This memory bank was then passed down to the bacterium's descendants, effectively immunizing entire future generations against that specific viral strain. It was a Lamarckian dream realized at the molecular level, an organism acquiring a trait during its lifetime and passing it directly to its offspring. While the academic world slowly digested the profound implications of Mojica's discovery, the first practical application of CRISPR occurred in a spectacularly unexpected arena: the global dairy industry. Companies manufacturing yogurt and cheese rely on massive vats of bacterial cultures, which are highly susceptible to catastrophic viral infections that can wipe out entire production lines in hours. Researchers at Doudna, analyzing the genomes of their commercial Streptococcus thermophilus strains, realized that the most resilient bacteria were actively using the CRISPR system to fend off bacteriophages. By intentionally exposing their cultures to specific viruses, they could force the bacteria to naturally update their CRISPR arrays, creating ultra-immune strains. Long before it was a medical marvel, ancient microscopic immunity was saving multi-million-dollar industrial food supplies. But the purely observational phase of this Genesis story was about to end. To understand how the bacteria actually weaponized these genetic mugshots, scientists had to look beyond the DNA arrays and examine the genes located immediately adjacent to them. These were the CRISPR-associated genes which produced complex enzymes known as Cas proteins. If the CRISPR array was the intelligence database, the Cas proteins were the executioners. When a recognized virus invaded, the bacterium transcribed the specific spacer DNA into a piece of RNA. This RNA molecule then physically bonded with a Cas protein, acting as a highly specific homing beacon. The elegance of this biological machinery is breathtaking. The RNA-guided Cas protein patrols the interior of the bacterial cell, constantly bumping into incoming genetic material. It checks the invading DNA against its RNA guide. If there is a perfect sequence match, the Cas protein undergoes a sudden conformational shift. It acts as a pair of molecular scissors, gripping the viral DNA double helix, unwinding it, and cleanly slicing through both strands, neutralizing the threat instantly. It is a highly targeted, programmable search and destroy mechanism, refined over billions of years of evolutionary warfare. The transition from a fascinating biological curiosity to a world-altering technology required a synthesis of distinct scientific disciplines. This pivotal shift began to crystallize in 2011 at a microbiology conference in Puerto Rico, where Emmanuelle Charpentier, a researcher studying the deadly human pathogen Streptococcus pyogenes, met Jennifer Doudna, a renowned structural biologist specializing in RNA architecture. Charpentier had recently discovered a crucial, previously unknown third component in the Streptococcus CRISPR system, a secondary molecule called tricRNA, which was essential for locking the guiding RNA into the Cas9 protein. Their collaboration sought to isolate and understand the exact biochemistry of this specific Cas9 system in a test tube, entirely removed from the living bacterial cell. They meticulously purified the Cas9 protein, the target-seeking CRISPR RNA, and the structural tricRNA. Through a series of elegant biochemical assays, they proved that these three components alone were entirely sufficient to locate and cut a specific DNA sequence in vitro. But the true leap of genius, the exact moment CRISPR transitioned from a natural phenomenon to a human-engineered tool, occurred when they looked at the two separate RNA molecules required for the system to function. In a landmark 2012 paper, Doudna, Charpentier, and their teams demonstrated that the two distinct RNA molecules could be artificially fused together into a single, continuous synthetic molecule, which they dubbed a single-guide RNA. They didn't just understand the microscopic machine, they had successfully built a customizable steering wheel for it. By simply swapping out the 20-letter genetic sequence at the tip of the single-guide RNA, they could program the Cas9 protein to seek out and cut absolutely any DNA sequence they desired. The intellectual shockwave of this realization rippled through the global scientific community instantly. The implications were staggering in their scope. For decades, genetic engineering had been a clumsy, imprecise, and incredibly expensive endeavor, akin to editing a massive encyclopedia with a blindfold and a pair of blunt shears. Suddenly, humanity possessed a molecular scalpel of unprecedented precision, a tool that was cheap, easy to synthesize, and capable of targeting any specific gene in any organism. The biological barrier had been permanently breached. The immediate engineering challenge was to prove that these bacterial scissors, evolved to operate in simple prokaryotic cells, could function within the vastly more complex environment of eukaryotic cells, specifically human cells. Eukaryotic DNA is not floating freely. It is tightly spooled around proteins and packed into a dense, heavily guarded nucleus. In early 2013, multiple research teams simultaneously demonstrated that the CRISPR-Cas9 system could indeed be imported into human cells, where it navigated the complex nuclear environment, found its programmed target, and executed a precise genetic cut. The bacterial defense mechanism had been successfully weaponized for human use. The floodgates of innovation burst open, unleashing a torrent of unforeseen applications that touched every corner of biological science. Within mere years, researchers were using CRISPR to engineer drought-resistant crops, to create malaria-resistant mosquitoes, and to systematically deactivate the genes responsible for hereditary blindness. In the realm of human medicine, it moved rapidly from laboratory theory to clinical reality, offering the first genuine cures for devastating genetic conditions like sickle cell anemia by precisely editing the flawed DNA within a patient's own bone marrow stem cells. The democratization of genetic engineering had arrived, moving power from massive, well-funded institutions to virtually any standard molecular biology lab. Yet, alongside the miraculous potential, a profound ethical shadow emerged. The extreme ease of use that made CRISPR so revolutionary also made it uniquely dangerous. The ability to easily alter the human germline, making genetic changes to embryos that would be inherited by all future generations, shifted from a theoretical bioethics debate to an immediate, tangible reality. We had gained the unprecedented power to direct our own evolution, to permanently edit the human blueprint. This godlike capability was not born from a deliberate, top-down engineering project designed to master humanity's fate, but was entirely derived from a single-celled organism's desperate struggle to survive a viral infection in a muddy puddle. This remains the ultimate paradox of the CRISPR genesis story. Humanity's most futuristic, powerful, and consequential technology was not invented from scratch. It was excavated. We are essentially riding on the coattails of more than 3 billion years of microscopic research and development. The precise, programmable molecular machines we now use to rewrite the code of life were perfected in the silent, invisible wars of the microbial world, long before the first animals ever crawled onto land. As we look toward a future where genetic diseases may be eradicated and the biosphere can be fundamentally reprogrammed, the echoes of that foundational discovery remain. The ongoing search for new genetic tools has shifted back to the microbial dark matter as scientists scour the genomes of bizarre extremophiles for other ancient defense mechanisms. The genesis of CRISPR serves as a profound reminder that the most revolutionary leaps in human capability often begin with the simple, obsessive curiosity to understand the strangest, most overlooked anomalies hidden within the natural world.